I'm joined here today by Dr. Corey Nislow from the University of British Columbia and Dr. Tim Hammond from Veteran Affairs in Duke. Gentlemen, we're here to talk today about Apex yeast. Why are we testing yeast on the ISS?
Well, yeast has actually been the organism or cell that's been domesticated for longer than any other. We have records dating back 10,000 years. And so essentially there's a lot of expertise surrounding working with yeast. And it happens that half of the genes in yeast are shared between you and I. So half of the genes in a yeast cell are just like yours and mine. And so there are experiments that we can do on yeast that you could never design or execute on a mammalian cell.
Mm-hmm. So what does that have to do with medical doctor and Veterans Affairs?
Well, in the VA, we have a very large number of diabetics, and some of the medicines we give patients for diabetes unexpectedly have very strong anti-cancer effects. So we're trying to work out the mechanisms by which these drugs can be repurposed to treat cancers in our veterans. And of course, that will carry over to US population and overseas.
So do you somewhat get your marching orders as to what they're trying to figure out with respect to the yeast and its correlation to human cells and how they work?
Well, marching orders is a good way to put it, although we tend to work very well together. The way our collaboration works is that Dr. Hammond's laboratory frames the experimental question, and it's almost always a question related to human health. Mm-hmm. And then we go back to the lab and design an experiment. Yeast are extremely flexible, and so we can use them and design an experiment that speaks to the question that Dr. Hammond is trying to address.
How closely related are the yeast to the human?
Well, evolution is a beautiful thing in that we split from yeast 800 million years ago, but half of our genes, the genes that control how our cells divide, are so well conserved that the same gene from a human cell can replace the gene in a yeast cell, and that yeast cell will operate just fine. So although we're a million years apart, functionally evolution has conserved those essential functions.
Millions of years apart, but we're also many miles apart between the Earth and in orbit in microgravity. How can you recreate what goes on in space and microgravity here on Earth to make the changes that you need as far as medical is concerned?
Well, it's actually pretty simple because in microgravity, gas doesn't convect very well. So if you have a gradient of gas, the gases don't mix like they would on the ground. And many people believe that that's what happens inside a tumor. So cells growing in space make a really good model system to study tumors on the ground. So if we find a pathway that's activated by a drug under those conditions of low gas, or as we call it, redox state in space, we can identify that pathway and use it therapeutically on the ground.
What is the end game if you find out the way that the tumors react and cells react in microgravity? What is the end game on Earth?
Well, it's actually really neat because these drugs have already been through all the testing with the FDA and have great safety data because they're used already for another purpose. So the purpose is to get a better anti-cancer drug and to perhaps prevent some forms of cancer, treat some forms of cancer, but if you know the right core molecule, you can perhaps design an even better molecule to fight cancer once you know the pathway that's being activated.
There's parallel testing that's going to take place here on the ISS and on a ground station. Can you tell me a little bit about that and why you do the tests at the same time or, within hours of each other?
Well, it's the principle of a well-controlled experiment. And I think Tim can probably speak to the details of the engineering behind why and how we do those controls.
Mm-hmm. So what we do is if we want to know if space is unique, it's exactly what you asked. Why do it in space if we can do it on the ground? So if we want to know if space is unique, we have to have a control on the ground to know that space is unique. So we actually do 3 experiments. We do one set static on the ground, we do one set in space, and we do a rotating set. because in rotation you can randomize the gravitational vector and reproduce some of the conditions in space. So part of this, the medical aim is to get a better drug. The practical aim is work out also when we really need space and when we can do things equally well or almost as well on the ground.
Now, I understand the instrument that's being used is from Canada to do some of the tests. Do you have any special knowledge, or have you done work with that prior to this program?
Well, we've run about 3,000 drugs against these particular yeast strains. And so we have a good baseline of data to which we can compare the space data when it comes in. And this is a very powerful tool. Some people call it big data. It's a way to take your results and ask sort of guilt by association. I can't interpret all of the data, but it looks most similar to this treatment. And then I can drill down and look at that particular treatment and get some new insights into how that particular treatment was affecting the cells.
Based on your research here, guesstimate maybe of how far down the road we might be able to see some, real world application of the results of this type of testing?
Well, a new drug takes decades to get to market. But remember, we're repurposing a drug that's already on the market. So it's going to be way shorter than that. How much shorter, I'll come back in a couple of years and tell you, because we may be overly optimistic, but it's certainly be much shorter than our new compound.
Well, it seems like the science and the research just keeps going and going and going, and we look forward to bigger and better things with APEX as you all continue to do good research. Gentlemen, I appreciate your time today.
Thank you very much.
We
Thank
look
you.
forward to your results.